Hi everyone, my research project was called Where Has All the Mass Gone? Quantifying TSS in Headwater Streams. I am Mackenzie Harpine and my faculty mentors were Dr. Lau and Dr. Stefaniak. So for a little introduction, the Mountain Valley Pipeline is a pipeline that will transport natural gas from West Virginia to Virginia, and it will span a total of 303 miles. They started building it in 2018, and as of June 2020, it was about 92% complete. It will cross a total of 1,000 bodies of water. They have not received permits that will allow them to cross streams in Virginia yet. The picture on the left shows a map of where the pipeline will be built, as you can see right here. And the picture on the right is a picture from a distant view from one of our sites. So the hole they have to dig to place the pipe is 9 feet across. The area they have to clear to install the pipeline will be a 125-foot easement of trees, which is equal to 38 meters. The pipe itself is 42 inches in diameter, which is equal to 1 meter. In this picture on the left, you can see the area that they have to knock down trees in order to dig the hole to build the pipe, or to place the pipe. And then in this picture labeled B, you can see how big and deep the hole is to actually place the pipeline. This picture to the far right shows the smallest stream that the pipeline will cross, which is 0.8 of a meter. The largest stream that the pipeline will cross is about 6 meters wide. So why will the pipeline construction process cause problems? This image shows a variation in the streambed habitat, and as you can see, there are bigger rocks as well as smaller sediments. And the construction process will introduce more sediment into the streams that will fill in sediment into holes that were once used as habitats. And the sediment will also be an issue for insects because their gills will be sheared off. And you can see their exposed gills here in the pictures to the right. You can really see it right here. So my research objectives were 1, to develop total suspended solid and total volatile solid quantification in water samples, and 2, to compare 2 seasons of total suspended solids and total volatile solids pre-pipeline. So here's a picture of our site map, and these sites are located in Southwest Virginia, just south of Roanoke. And this dotted line here is the proposed pipeline pathway. And as you can see over here on the left, Site 10 is not affected at all, and it is considered to be one of our control sites. Along with Site 10, more controlled sites are here in black, which you can see Site 1, Site 4, Site 9, and Site 6. And then our impact sites are here in red, which you can see is 2, 3, 5, 7, and 8. So what does this all mean? I specifically quantified total suspended solids and total volatile solids before the Mountain Valley Pipeline in Roanoke was built. My research was focused on the total suspended solids in the water, which is matter that is not yet dissolved into the water column and can be trapped into a filter. Total volatile solids are organic matter in the water column that can be burned. I looked at the difference in the water between the spring 2020 season and the spring 2021 seasons. This project is ongoing, and after this pipeline is built, a student can continue to use my protocol to continue to quantify TSS and TVS in these streams. So, what is the problem that we face? So previous students could see the sample on the filter papers after filtering the water samples, but they were getting negative numbers. The weight of the filter paper was 125 milligrams on average. When prepping the filter at 105 degrees Celsius, they found that on average 0.47 milligrams was lost. They knew that they were losing mass after filter preparation, and they were losing 1 milligram after using the muffle furnace. For my research, we decided to prepare the filters at 500 degrees Celsius instead of 105 degrees Celsius because the muffle furnace shrinks the filter paper. We also decided to filter whole samples through one filter paper instead of just 500 milliliters. Now that we fixed these problems, we can get into the new methods of my research project. So in total, I looked at 10 samples from spring 2020 and 10 total samples from spring 2021. So all in all, I looked at a total of 20 samples. To begin, I had to weigh the filter paper with the weigh boat and then weigh the weigh boat separately. Then to prepare the filters, I washed them each with 100 milliliters of deionized water. After the deionized water was filtered through, I placed the filter paper and weigh boat in the muffle furnace at 500 degrees Celsius for 1.5 hours. In this picture on the left, this is what the muffle furnace looks like on the inside. And then the next step, I cooled them in the desiccator, which you can see here in the middle picture. And in this picture to the far right is the vacuum apparatus that I used. So this was the cup where the water would sit. And in this blue piece is where the filter paper would go. And it was hooked to a vacuum that would pull the water through, and the filter paper would collect our TSS. So for the methods continued, I washed each filter paper with a water sample collected from streams around where the pipeline will go. And this, you can see here in this left picture, is the amount of sediment that was collected, TSS that was collected in this filter paper. And then after that, the samples were filtered. The weigh boat and filter papers were placed in the oven at 105 degrees Celsius overnight. And this middle picture shows the inside of the oven. Then the next day, the weigh boat and filter paper were weighed again to get the final total of TSS, which is shown in the picture on the right. So right here. Then after they were weighed, they were placed in the muffle furnace at 500 degrees Celsius for another 1.5 hours. After they were cooled in the desiccator, they were weighed again, and this was to find the total volatile solids. Then just the weigh boat was weighed because we found that the filter paper was sticking to the weigh boat. And we found that on average, the weigh boat gained about 0.07 grams. So after we solved the problem, here is our completed data. So in these pictures, you can see the final results of the filter papers. As you can see, some are darker than others, which means there was a lot more TSS in that water sample. These show that there is a visual representation of how each site varies. So now that I explained the methods, I am going to show you some of the results. I performed a paired t-test because we have repeated samples per site. This figure compares the spring 2020 season versus the spring 2021 season and looks at the TSS concentration. So here on the x-axis is where you can see it compares the seasons. And here on the y-axis, you can see it is the TSS concentration in milligrams per liters. Although the mean difference is 3.3, which you can see right here, the difference is not considered to be significant. Next, I looked at TVS. So the TVS concentration in spring 2020 is significantly higher than spring 2021, which you can see here on the left. And finally, I looked at the ratio of TSS to TVS. So this ratio shows the TSS to TVS, and it was significantly higher in spring 2021 than in spring 2020, and you can see it here on the right. So this mean shows that there is more TSS relative to TVS in the spring season of 2021. We concluded that there was a higher ratio of TSS to TVS in spring 2021. And we think weather had an impact maybe on the spring 2021 data because it rained quite a bit the days prior to when we collected the spring 2021 water samples, and it may have stirred up the materials in the water. Once they're able to cross the water in Virginia for the pipeline, TSS is expected to be higher than what we found pre-construction, and TVS is expected to decrease post-construction due to the removal of the trees surrounding the area. This is continued research and lays a background for further research. So I would like to thank the Jeffers Trust Award. I would also like to thank the biology and chemistry departments for allowing me to have a place in the equipment to do my research. I'd also like to thank Dr. Stefaniak for trusting me to continue this research. I'd also like to thank Dr. Lau for helping me through this process and guiding me along through my tons of questions. I would also like to thank the landowners for letting us collect data on their property. Also, a big thank you to the Office of Undergraduate Research for allowing us to present and for providing us a platform to present our work. And last, a huge thank you to all of the students and professors who have helped collect samples and data for this ongoing research. So, does anyone have any questions? Thank you.